Shown here are the reagents and tools needed for DNA extraction and isolation. For this project, we suggest using one of two DNA extraction and isolation methods. The two methods discussed here are the Chelex and the hot shot method. The first extraction method, Chelex extraction, involves warming a sample in a 5% suspension of deionized water and Chelex 100 resin. Chelex has an increased affinity for heavy metal cations, including magnesium. Magnesium is a necessary cofactor for nucleus activation, and binding these ions inactivates the enzyme. The alkalinity of the suspension in the boiling process lyses the cell membranes and denatures cell proteins. The suspension is then centrifuged, separating the resin and the cellular debris from the supernatant containing the DNA. This purified DNA can then be used in downstream applications such as PCR amplification. It is important that the Chelex beads never make it into our PCR reactions, as they will disrupt Taq polymerase. Chelex is recommended for soft tissue samples such as fungi, buccal cells, or soft plant tissue. Before you begin the Chelex extraction, turn on a heating block or water bath set to 95 degrees Celsius. To prepare a 6% Chelex solution, mix 3 grams of Chelex with 50 milliliters of deionized water. Mix thoroughly by lightly shaking the solution until a uniform mixture is apparent and no visible clumps are present. For each sample, aliquot 200 microliters Chelex solution into a 1.5 milliliter microcentrifuge tube. Swirl the solution in between each aliquot to keep Chelex beads suspended, as they have a tendency to settle out of suspension. It is best to use a 1000 microliter micropipette to dispense the Chelex solution, as the diameter opening of the smaller pipette tips may be too small for the Chelex resin. If you are having trouble with your tips clogging during this process, you can cut the tip to increase the diameter. Prior to transferring samples to the tubes, be sure to label each tube to correspond to your sample ID listed on your collection worksheet. Label the tubes on both the cap and on the side. Keep labels legible. Dip forceps into a 10% bleach solution and wipe clean with the sterile Kim wipe. Allow the forceps to air dry for approximately 5 seconds. Using the sterile forceps, remove a small piece of sample tissue. Remember, the piece of tissue should be extremely small and just barely visible, as too much tissue may inhibit your reactions. For example, with insect samples, if the sample is larger than 5 millimeters in total size, use a 1 to 2 millimeter piece of the abdomen. If the whole insect is smaller than 2 millimeters in total size, use the whole insect. Be sure to research the appropriate amount needed for your type of sample. Once you have removed the piece of sample, uncap the tube of Chelex, place sample in the appropriate labeled tube, and close lid. Tougher materials such as pine needles, or if your tissue sample is tough, you can manually grind the sample with a toothpick or small pipette tip to help break down the sample. Or you can use a mortar and pestle before transferring the sample to the Chelex. Repeat with each sample in a new Chelex tube, being sure to sterilize forceps between samples. When finished with all tubes, either manually finger flick or vortex samples in Chelex slurry for 10 to 15 seconds. Be sure lids are snapped on tightly before beginning the heat incubation. Spin samples briefly, 10 to 15 seconds, at maximum speed in a microcentrifuge. This step is to ensure that the sample is inside the slurry of Chelex. Incubate samples for 20 minutes at 95 degrees Celsius. The block temperature may drop slightly when doing this step. This drop is normal. Check tubes while incubating to ensure that lids have not popped off. Vortex samples again for 10 to 15 seconds. Be careful, as steam may pop lid off of centrifuge tube. Spin one last time for 5 minutes in centrifuge. The sample is now ready for use, and the DNA will be located in the supernatant, while the cellular debris will be mixed within the Chelex pellet at the bottom of the tube. The extract can be stored at 4 degrees Celsius for up to 1 week. The second extraction method is referred to as the hot shot extraction. In this extraction, an alkaline lysis solution containing sodium hydroxide in combination with heat is used to destroy the cell membrane, contaminating proteins and DNases. The lysate is then neutralized with a secondary solution containing tris to bring the pH to a level conducive to performing PCR. This method is very straightforward, but does require you to prepare an alkaline lysis solution and a neutralization solution ahead of time. Also, before you begin the hotshot extraction, turn on a heating block or water bath set to 95 degrees Celsius. Label each tube to correspond to your sample listed on your collection worksheet. Label the tubes on both the side and on the cap. Put as much information as you can manage, but always keep labels legible. Adding initials and a date is also a good idea. Cut a 1 to 2 millimeter piece of tissue and place in a 1.5 milliliter microcentrifuge tube. If using insects, once again use a 1 to 2 millimeter piece of the abdomen if the insect is larger than 5 millimeters in size. Or if the whole insect is smaller than 2 millimeters in size, use the entire insect. Once again, caution, larger pieces can inhibit the PCR reaction. Add 100 microliters of alkaline lysis reagent. Ensure that the tissue is completely submerged. Vortex or shake vigorously for 1 minute. Make sure that the sample is completely submerged in the lysis solution. Add 100 microliters of the neutralization reagent using a new tip for each sample. Mix well or vortex, or use pipette tip to break up sample. Sample may be thick and discolored. This is normal. Though not necessary, you can centrifuge the sample to pellet the debris. Similar to the calyx extraction, the DNA will be in the supernatant. At this point, the extract can be stored at 4 degrees Celsius for up to a week, but it is best to use it within a few days.